This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 602, recorded on April 14th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hello there, Vincent. How are you today?
I'm pretty well. Yesterday we had a huge storm. We lost power throughout the day. But
is that right?
Nice today. Yeah. It's pretty weird. We have a pandemic. We have a power outage.
We have a little bit of a cold snap here. It was 45 this morning. I'm just checking to see what it's. High is supposed to be it's 58. No, that's Washington. 64 right now in Austin. Fair enough.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It's almost as warm as it is in Texas. It's 56 today. And it's nice and sunny after that big rainstorm we had the last couple days.
Did you have power outage yesterday?
I actually did not have a power outage yesterday, which is strange because I get power outages easily.
Yeah, we had multiple. I actually set up a generator so I could teach my course uninterrupted, you know?
Nice. That's dedication.
And yeah, because earlier in the day I was editing things that kept going out. And I said, the hell with this. And I left even after the power came on. Kathy Spindler will be joining us shortly, but we have a guest today from the University of Iowa, Carver College of Medicine. He is a professor of microbiology and immunology and pediatrics. He is returning for the second time to TWiV. Stanley Perlman, welcome back to TWiV.
Thank you, Vince. Good to speak to you, Rich and Brianne.
Well, you were on TWiV last year. We came to visit you in Iowa. March 10th, 2019, it was released, TWiV 538, and the name, listen to this name, an Iowa Caucus of Viruses.
A long time ago. A long time ago. Yeah.
It's a long time in many ways. Of course, the caucus has passed, and we don't want to talk about that. Now we have a pandemic to talk about, and we want to talk about the immunology of SARS-CoV-2 and other coronaviruses with you. So I'm sure it's something you've thought about a little.
Yes.
Stanley, you're one of these guys who have been laboring away in obscurity for your whole life until first SARS and then MERS and now SARS-CoV-2 happens. And I don't think it's going away this time. I mean, sure, the pandemic will be over, but coronaviruses are going to be on everybody's radar forever now. So,
yeah, that's what I think too.
Yeah.
I like this. You wrote a commentary in the New England Journal early on. You called it Another Decade, Another Coronavirus, right?
Yes.
I like that very much. I didn't realize every 10 years now, right?
Well, we hope the next one lasts 10 years without another one.
That's a good question, right? Who knows? Anyway, let's talk about the immunology of... So let's start with SARS coronavirus, which, people call classic SARS now. Probably you know that.
Yes.
What do we know about the immunology? You've done a lot of work on that. Can you dissect it for us?
Yeah, so maybe we can go back even earlier to the common cold coronaviruses.
Absolutely.
Because these were discovered in about 1960, and they were found to cause the common cold. And studies were done then on volunteers because volunteer studies were allowed then or more favorable. So they took people who had been infected with the cold coronavirus, and sometimes experimentally and sometimes naturally, and they measured antibody titers, and they discovered that there was signs that the person had been infected previously, and then they went and gave these people the same virus, and lo and behold, they all got colds. There was there's a variable degree of a cold, so some people that seemed to have low proteins to the antibodies, low antibodies in the blood, had a cold that was milder than those who had low amounts. So if you had a high amount of antibody, I think I said that backwards. A high amount of antibody, you got a more mild cold. If you had a low amount, you got a more severe cold. So this told us even from back then that this was behaving what we thought cold viruses were doing anyway, which is that they gave you an infection. It was a mild infection. You had a mild immune response. It didn't last too long. And then you could get the same infection again with plus or minus the same degree of symptoms. So that's
there was no cross-reactivity between the different common cold coronas, right?
That's right. So there was no cross-reactivity and between the common ones. And at that point, there were only two. One was called 229E. The other one was OC43, which were themselves interesting viruses because one of them, OC43, may have come over from cows. So it's almost identical to a virus that infects cows, while 229E was probably an ancient bat virus from 1,000 years ago. And it's been found in camels, actually, in the Middle East now. And for all we know, this caused a bad set of diseases 1,000 years ago. We don't know, of course.
When would that cow spillover have been?
We don't know. It occurred, from studies, it looks like it occurred about, 100 years ago. But we really don't know where it occurred. But what we do have evidence for is cows infecting other ruminants. So other animals that are like cows can get infected with this virus. This virus is very non-discriminatory. It's quite promiscuous because unlike the viruses that we're going to talk about in a few minutes, this one seems to bind to sugars to enter cells. So its requirements for entry are not great. So it's really a great virus for spreading around from not only person to person across species but it causes a common cold and occasionally and rarely more than that.
Do we understand why the antibody responses are not great or why they wane, I guess, is the real question?
Yeah, so I don't think we understand that. And that's going to be a theme of SARS and MERS too, particularly MERS, which I know more about. We were just dealing with these two human cold coronaviruses, and both of them were not really easily studied because one of them really couldn't infect mice. We made mouse models that should have worked and they just didn't work. And in addition, mice don't get colds. So it's really hard to study the common cold in a mouse. So that was one of them and never did much of anything. The one that bound to sugars caused mostly a brain infection. So it really liked to go to the brain and cause disease that had nothing to do with an upper respiratory tract infection. So that's where we stood in about 2000 in terms of human coronaviruses. We didn't have great models. We knew a little bit about them, and they would cause the cold, so they weren't highly studied.
So you've mentioned. Sorry. You'd mentioned antibody responses. Did we know much about any other immune responses, like T cell responses or innate cytokine responses?
Well, prior to 2003, I would have guessed we knew nothing about T cell responses, and we knew probably something about cytokine responses. I don't think they were different from other colds, which involve cytokines like IL-8 to bring in neutrophils. We know the mechanism of colds, and these coronaviruses didn't appear to be any different than any of the other viruses that cause cold, which of course are largely rhinoviruses, Vince's group of viruses. So maybe not his favorite one. So that's all we knew really I would say in 2003. So we knew, what we did know more about is that, there was a lot of, interest in, and still is, in about coronaviruses that cause disease in the companion animals and domestic animals. And a lot of these are really important. So one of these was transmissible gastroenteritis virus. And this was a virus that caused disease in pigs. It was important in my state in Iowa because it caused great economic loss because baby pigs got infected with this virus and had diarrhea. There was a disease related actually to the whole human cold coronavirus, the one that came from cows. It actually caused respiratory disease in cows, particularly when cows were all thrown together in a cattle car. So they all spread the infection among each other. It also caused, I think, diarrhea in baby calves. And then there was diseases in dogs and cats that were also important too. So that's almost where more of our information came from, and vaccines that were developed against those kinds of infections.
So those animal infections, do they show the same lack of durable immunity that you described for the common cold coronas?
It's a good question because a lot of them are for animals that are farmed, so they didn't live very long. So I don't know. I don't know what I can tell you about TGEV, because that's the most interesting one. So that was a terrible disease. There were vaccines that were made that were not very effective. I can't tell you why they weren't very effective exactly, but what I do know is that about 20 or 30 years ago, nature decided that he or she had had enough of this, and a new coronavirus appeared on the horizon that still could infect the lungs, not the lungs, the upper airway of baby pigs, but no longer infected the gut. So they no longer got diarrhea, and pigs basically were immunized. So TGEV disappeared. And so much so that my colleagues who worked on TGEV had to change viruses because there was no TGEV around. So it was really amazing. So a small change in the surface glycoprotein, WAMO. The virus is no longer in pig populations.
Let me welcome Kathy Spindler. Hello.
Hi, everybody. It's nice to be here.
Good to have you.
I zoomed from one meeting to another here.
The Zoom life.
Hi, Kathy.
Hi, Stanley.
So the fact, Stanley, that the TGEV, you said it immunized pigs after it changed. So that implies that you can make a vaccine against some of these viruses at least, right?
Right. And that's a model for the kind of vaccine that we know works well, a live attenuated vaccine, because it basically was the whole virus just missing parts of it that made it have a different tissue tropism or different tendency to bind to different organs in the body. And because of that, it didn't cause the disease that we cared about, but still infected the pig enough to allow an immune response to develop.
Aside from this natural vaccine for TGEV, not really a vaccine, but were there any other animal vaccines that were effective?
There's some that were partly affected but had other issues. So there was one for chicken called Infectious Bronchitis virus, and there was a vaccine for it. And here again, a live attenuated vaccine was used. But this time the problem was that I think there were, and again, I'm not a complete expert on this, but I think there were several avian vaccines against this virus in chickens. And what they did is they were actually excreted, much like other live attenuated viruses are excreted like polio virus. And the result was that these live attenuated vaccines all recombined, and you ended up with new strains of infectious bronchitis virus, which caused disease. So it was a less than ideal situation. I don't know what the status is right now with these vaccines. There may be a better one. But the last time I checked, this was what the story was.
That sounds like much less than ideal.
Yeah, it was much less than ideal. Chickens are funny because they don't have to live very long. So I'm not sure how this all worked or how it played out. And of course, chickens are often raised in these very close quarters. So it makes spread of any virus that you get really easy, just like in that shipping fever that the cows got when the cows were all together in a cattle car.
So, yeah, it sounds like the Marex vaccine, which doesn't actually prevent infection of chickens, but allows evolution of increased virulence or increased transmissibility.
Yeah, bad idea.
So I was going to ask you if you think that the infectious attenuated vaccine would be the one that works for SARS-CoV-2 based on the TGEV story.
Yeah, so I think that people have been thinking that ever since SARS and then MERS and then COVID-19, that might be the best vaccine. But there's always the caveats of getting a vaccine that's safe and is not going to mutate back to the more virulent strain. Yeah. And so there's just a lot of issues. There's even discussion now, even a vaccine that's as useful as measles, would it be approved now as it was in the 1960s or 70s? Because it's a live attenuated vaccine. Clearly measles is a great vaccine. So but this has been one of the issues. People are developing live attenuated. I don't know if they'll get traction though for going into human populations.
It's interesting because, there's a new polio vaccine that's been developed for type 2 that may be approved by WHO in a couple of months. Supposedly doesn't revert as much as the other one, but maybe there's a precedent there already, and we're in a situation where we have to prevent circulation of the vaccine-derived strains. So This
Right.
would be the only solution to that. So the common cold and the animal coronas, what about SARS-1?
Yeah, so SARS-1, so this was, of course, a disease that appeared maybe November 2002 and then was eliminated by July 2003. During that interval, we learned a lot about coronaviruses. A lot of scientists entered the field, and our knowledge increased greatly. And several vaccines were starting to be developed. And some of the work was done with some was done with live attenuated vaccines. I don't think they got very far. But what was more popular was taking a whole virus and inactivating it, and then using that with an adjuvant, which is something that enhances the immune response. And the idea was to use that for protection. There were also vaccines that consisted mostly of the surface glycoprotein to induce antibody responses. So these were almost to the stage of testing. There was one of the latter ones, one was the one where we had just the spike protein, was actually going to be tested at the University of Iowa because we have a big vaccine testing center. Mm-hmm. And then testing stopped because there were some reports of vaccine side effects. And it wasn't quite side effects. I would want to use the words immunopathology, which means that when you look, and this was done in mice and monkeys, when you look in their lungs, you saw differences from normal immune response. You didn't see a difference in clinical disease, and in some cases, you actually got some protection. But this was felt to be worrisome enough, particularly for a virus that no longer existed, that the studies were halted.
This was for both the spike and the inactivated vaccine?
Yeah, so for both of them, the different studies, they weren't all done with everything wasn't done in parallel, but yes, both of them had similar issues of either in one case, it was more an eosinophilic response, which is a sign of an allergic response. And the other one, there was a change in the type of macrophage, which is, of course, important for many functions in the lungs. And so both of these didn't affect clinical disease. The animals were mild, didn't have as much disease before and didn't have much disease after, plus or minus the vaccination, but after challenge. So it didn't matter clinically, but it was enough of a concern. And with all the worries we have about vaccines wanting to be perfectly safe, these vaccines were no longer studied.
So were there any sort of studies in humans just to look at immune responses, or were they all stopped sort of even before a general immunogenicity study?
Yeah, that's a good question. I know we stopped before any patient received anything or any subject received anything. I don't know if other centers went a little further than that. But as far as I know, there was nothing that went especially far. And there may be some that I'm missing from 10 or 15 years ago. But there was nothing, it wasn't like there was anything that we can hang our hat on when this virus came around and we could say, boy, that vaccine was great. We had tested it. We knew it was working just fine. And let's use that as a model.
So many of the vaccines that are being developed now rely on just the spike protein. Do you have a perspective on where that might lead us, whether that's likely to be an effective strategy or whether there are problems associated with that?
Yeah, so that's the question, because we know that a spike protein, especially in some formulations, will give you primarily an antibody response. And we know that for the most part, except in rare exceptions like polio, you need more than an antibody response in order to have protection that's durable and effective. So if you have the spike protein and it's delivered in a way that only gives an antibody response, you may have a response that wanes more quickly or isn't as effective as you like. So that's why a lot of us think that having a T-cell response is really critical to have along with the antibody response. And of course, T-cells are the cells that actually remove infected cells as opposed to antibodies, which tend to more remove the virus that's floating around in the blood or other places free from the cell. So we think that both are important. I think it really matters what kind of vector you use to deliver the protein in question. So whether you need others, we know that the T cell response in a lot of these viruses is directed at the spike glycoprotein, but it's also directed at one or two of the internal proteins. So to me, it's less whether you include the spike and other proteins, and more whether you can have a good immune response against both. That's both antibodies and these T cells.
Is there any evidence one way or another as to whether proteins other than the spike protein have a role in conferring immunity?
Well, we know that at the antibody level, those proteins, there's often lots of antibodies to them, but we don't really know that those are very effective in protection. We know that T cell responses can be directed against any protein. Even the small ones that are there in small amounts can have a T cell response. Whether one part of the T cell response is more important than others, we don't know. We know from HIV that there were some HLA types that did better with HIV than others. So the type of T cell response may matter. Whether it's the specific epitope or something else, I think is not totally clear. But we know that can matter in the kind of response you get.
So in my sort of childlike, my naive conception of immunology, thinking about this, whether you get exclusively an antibody response or a T cell response as well, I feel like if you just administer a vaccine as a protein already made, that you're likely to get a response skewed towards just an antibody response. Whereas if you administer a vaccine where the protein is actually manufactured in the cells, then you're more likely to get both an antibody and a cellular response. Is that a fair description?
Yeah, I think that's a fair description. In fact, for MERS, the two vaccines made it to phase 1 trials. One has been published, one is about to be published. And the one that made it to clinical trials was a DNA vaccine.
Okay.
So that's taken up by cells, and that's one that induced both antibody and T cell responses. One of the issues with that study is that the neutralizing antibody, which is what prevents this virus from getting into cells, that one was present a few weeks after immunization, but by a year it was largely gone from the people who had been immunized. So pointing out the difficulties in vaccination, because this was not a long-lived antibody response. T-cell responses also in this particular study waned by a year after vaccination.
So in those studies, is there any sort of follow-up to assess whether there's memory? So yeah, the antibodies or T-cells may wane, but maybe there's sufficient memory so that on re-exposure to the pathogen or in a boost, you would get a quick and robust immune response.
Yes, that's a really good question, because of course what we care about is not a measurement in the blood, but what happens when you then see whatever you were immunizing against. And I don't think we know the answer to that. One of the issues with both SARS and MERS, so SARS, or SARS-1 or whatever we're calling it, classic SARS, is gone. So there's no chance for that really to be tested. MERS, we're at the level of about 2,500 cases in 8 years, 2 a week. So it's the odds of someone, and they're all in Saudi Arabia, which has about 25 million people. And if at 2 a week and 25 million people, the odds of someone getting infected twice was really bad luck. So we don't really have a test for whether there's protective immunity, or if we could even detect it, because of course, if that person is protected, we don't even know he got infected the second time.
There is a camel MERS vaccine, though, right?
No more advanced than the human one. So the idea was to make a vaccine against MERS in camels, with the idea being, if we could prevent MERS in camels, maybe we would prevent the human infection. So you can do this. The study I know best really only looked at camels for about 4 to 6 weeks. And there was a good immune response to the vaccine. But camels are really tricky because, first of all, camels all over Africa and Middle East and parts of Asia are infected with MERS coronavirus. And they seem to get reinfected because they all have good antibody titers. So we think they get boosted periodically, sort of like a cold virus. Camels get a cold with this virus. They get a cold when their babies are born and the babies become susceptible. Babies get sick just like people. Send their child to a daycare center. They can get reinfected with things they saw when they were 3 years old, and they get a nasty cold. Well, the camels get the nasty recurrence of their cold, and that's when they have lots of virus and spread it to other young camels and to people. People happen to be around when the camels have their runny nose.
So speaking of boosting, by the same token as not knowing whether there's memory because there's not enough virus infection around to test it, we don't know whether if there were repeated boosting, whether that would be protective. It seems like the boosting idea is being floated as if the SARS coronavirus 2 develops into something that's more like the common cold coronaviruses, and we get repeatedly boosted with them by exposure to kids who are asymptomatic, that might provide protection. But what are your feelings about that?
Yeah, so this is really a crystal ball question, because if the virus, so what's the goal here? So to my mind, the goal is if you get rid of the severe pneumonia, then this basically turns into a really nasty cold. And, a little more, people feel pretty rotten with it, even if they don't get pneumonia. But it's a disease that's tolerated by most people. It would probably still be a disease that would be a problem for aged individuals. So not a totally good outcome, but better than having severe pneumonia. So if we had some sort of herd immunity where a lot of people are protected, or we do a vaccine and we get rid of the pneumonia, that's one step forward. Then the next step forward is, can we minimize transmission? Well, minimizing transmission matters if a disease is severe. The reason, as I think Vince knows best, with rhinoviruses, we had a drug that worked against rhinoviruses, and the problem was that no one really wanted to buy it. It was hard to figure out when to exactly use it, but no one wanted to pay money to prevent a cold. So the drug did not do well, and it's been taken off the market. So the same thing that we could reach that point here where the deep pneumonia is very rare, or the severe pneumonia is rare, and we have a cold that's nasty, maybe gets less nasty with time. And, at that point, yes, there's going to be boosting, and it will not be a big deal for humans. This is, of course, not anywhere given. It may be that this virus doesn't change into just a cold virus, that immunity doesn't develop in a way that prevents the severe pneumonia. These are things that we don't know, but it's certainly a possibility that you have enough immunity so the amount of virus you get in your nose or airway is less. Because of that, you get less down to your lungs, and most people don't develop anything close to the severe pneumonia that we're seeing now. But there's still transmission at a level that may be equivalent to what we're seeing now or a little less. So these are the kinds of variables we have to think about. I think thinking about sterilizing immunity is probably a bad idea. If we're thinking about getting rid of this virus completely, it's not that it's a bad idea to have that as a goal, but it's a bad idea to be dissatisfied if we haven't reached that goal.
So there have been some concerns about antibody-dependent enhancement, and I don't really understand the concern. But you have published a paper which suggests that antibodies against IgG, the spike IgG, can cause severe lung injury. In fact, that's the title of the paper. So, can you explain the current concern about this in SARS-CoV-2 infections and that?
Yeah, so let me go back to the paper a second, because this is one of the when we were talking before about what vaccines could do. So this is one of the papers that's illustrated as causing vaccine-dependent damage to the lungs. But this is a paper where the macaques did have changes in what their lungs looked like when you examine them on autopsy, but clinically they had no change. So no matter what the lungs look like and whether you call them severe or not severe, and there were changes. The animals themselves didn't care because they clinically had no difference in their disease. So this to me is one of the important things to consider in this whole discussion, because you don't want to eliminate good vaccines because there's abnormalities on looking in the lungs under the microscope. But on the other hand, if these microscopic changes translate into changes in clinical disease, especially worsened disease, then of course we care. So this is an issue that with that paper, because it certainly raised a lot of questions. So antibody enhancement. So I'm at a committee actually in fact, two committees that are dealing with this. And what we're calling it is actually vaccine enhancement, because for antibody enhancement per se, it's a little more nebulous about whether that's the only way you can get this kind of change in cellular infiltrates. So, for example, in the past, when people have talked about antibody enhancement, they've talked about what occurs in dengue virus infections, where you can have an antibody that's suboptimal, and the virus can now bind that antibody, and instead of being neutralized, so they're being eliminated, in fact, it's now able to infect cells that it couldn't infect before, so you get increased infection. So that's antibody enhancement. It's usually of a cell called a macrophage, which we talked about a little earlier. So that's one kind, and we don't see that in SARS or any coronavirus infection, except there was one case where cats were immunized in a way that they had illustrations of this. But there's never been any other report of this particular kind of problem occurring after a vaccine or a natural infection. So that brings us to a second kind of vaccine problem, which is that you get a difference in the kinds of cells that go into the lungs. I think we mentioned this a little earlier. And here we're talking about cells, the composition changing. So you have the eosinophils, which are the signs of an allergic reaction, which I think I mentioned earlier as well. And so these, we don't know, we don't want this as in the lungs of people who are immunized. Whether this is actually a sign of a problem, I think is not completely known. But we're trying as much as possible, of course, not to have any of these things occur. And we think by having a balanced response that includes both antibodies and T cells, as we discussed a little bit, that it's likely you won't have these kinds of problems after vaccination. But that's really what vaccine enhancement is right now. It's really trying to define how are we going to even see if somebody has vaccine enhancement, because this is a serious disease of variable penetration, as it were. So people get colds, people get nothing, people get severe pneumonia. So if they were vaccinated, how would you tell vaccine failure from somebody who's actually had enhancement of disease? So that's why there's a discussion about it, because it's not so easy to figure out the best way to measure it.
So these changes in the lung that you described in the macaques or in these other circumstances, are they transient? In other words, or would it clear up eventually, or is this a more lasting thing?
I don't think that's ever been studied that way. My guess is it would clear up, because even within the macaques, the amount of virus was decreased. So these cells were there, but I'm sure they were going to go away with time. And I don't know, maybe we should have asked what happens if if we look longer, are we going to have changes in the lungs in terms of healing that are worse after vaccination? That wasn't done as part of the study though. So, but that's a good question about whether they would go away. My gut feeling is that they would resolve, because I haven't seen evidence that they really are causing major lung destruction, which is why you usually have problems with your lungs after an infection is cleared. It looked to me just like it was cells that shouldn't have been there that were there. So, but it's a good question to know what would happen long term here, and we don't know the answer to that.
And it's a phenomenon that you're not really going to be able to study in humans, right? Because it's not like you can get the appropriate lung samples easily.
Yeah, so you may not need to, though, because if coming back to the issue of how important it is, if you have somebody who has this vaccine-enhanced disease, if we know that people who are vaccinated now are coming back with the same spectrum of disease as people who weren't vaccinated, mild, severe, and that the people who had maybe moderate disease now are coming back with long-term lung problems, then you can say, well, maybe that's something to do with vaccination. A lot of the things that we know about vaccine-enhanced disease, so for example, with the killed measles and the killed RSV vaccines in the 1960s, it wasn't like that was apparent immediately. It took a while for that to filter through, for people to see what the kinds of problems were.
Yeah, unfortunately, that's the kind of thing you don't necessarily see until you've actually licensed a vaccine and used it in a lot of people for a period of time.
Yeah, that's right. And that's of course, that's the reason that people are working so hard now to define vaccine enhancement, because we don't want to have that happen again. When, we're not taking the attitude of, time will tell what'll happen. We try to do the best we can to make sure that we're doing something where there's none of this damage done so that there's not a problem. And that's why we're thinking about it. It's just hard to really assess it.
This range in severity that you mentioned, from mild to serious, do you see this with SARS-1 and MERS infections as well?
SARS-1, the idea was, the thinking was that everybody who got infected was symptomatic, but there were clearly people who had more severe disease and people who had less severe disease. I don't think there was any reports of people who were asymptomatic or had subclinical disease, but it's a little hard to know because the studies weren't done quite as I don't know, maybe aggressively as we're doing now. With SARS, people who were taking care of these patients had fewer resources in China and Hong Kong, and they were mostly worried about saving lives and taking care of people. So they didn't, I think someone who had mild infection might not have been diagnosed as much, because maybe I would guess that people didn't care so much. We know there's one hint of mild disease, and this may or may not be relevant. So, SARS, the way it crossed to humans, it was originally a bat virus that was in the live exotic animal markets in Guangdong province. And when it crossed over to people, it went through palm civet cats and Chinese ferret badgers and raccoon dogs. And when it went through these animals, it adapted to those animals. And we could see that mostly in the spike glycoprotein. And when it then crossed over to people, we could then again see changes in the spike glycoprotein that allowed it to infect people better. But we know that in the live animal markets, something like a third of the people who worked there had antibodies to SARS-CoV-1, and which means that they were infected and they got some mild disease. And of course, the virus wasn't adapted, so that's why it may not be relevant to make statements about subclinical disease. But so we know that some people could get infected with pretty mild symptoms. And I would bet the same thing was true in SARS-CoV-1, but it wasn't really studied to a great extent. And in MERS, we have more information because we and others have found patients who have almost no disease who, well, let's say we're in the hospital. This is a lot of, because MERS is mostly a hospital disease. So people were in the hospital and the healthcare worker wouldn't take proper precautions in the beginning. And so some of them would become infected. A lot of them had really mild disease and barely developed an antibody response. And if they did develop it, it would often go away. So we know that mild disease occurs there as well.
Do you think it's occurring also in SARS-CoV-2 patients?
Mild disease?
Mild or asymptomatic, yeah.
Oh, yeah. I think there's lots of evidence for some proportion of people having it. And it's really, the range that I read is just overwhelming because some people say just a minority of the total cases. Then other people say it's 10 times as many cases because of the way the epidemiology is looking.
Yeah, I think WHO has said that asymptomatic infections have no contribution to transmission. I don't know how they can say that, though.
Yeah, I think it's, my feeling when I think about colds, especially as a pediatric infectious disease person, until this all began, I would have said that if you don't cough and you don't sneeze, it's really unlikely you're going to spread virus very much. But in fact, if there's people who walk around with lots of virus in their nose and no disease, then the idea is that maybe some of them can spread virus by very small particles from breathing or talking, and that these could spread. What I would say in support of what the WHO says, it seems like this is a minority of transmission, even though there's evidence of people saying, I wasn't around anyone and here I am getting sick. And, we all also remember, if you think about our normal lives before all this began, there was all of us, I think once a winter, everybody would come home and say, I just got a cold or I just got some infection and I wasn't around anyone who was sick. So how in the world did this happen? So you really wonder.
So there are also some reports of people getting reinfected shortly after being released. And I don't know what to make of those. Can you shed some light?
Yeah, I think that I have the same questions you do. I think that they're probably not reinfected. Could people get reinfected? I suppose it's theoretically possible. I would have thought there'd been enough of an immune response so that it never occurred. So I think it's more likely that you're dealing with low levels of virus or viral products. See, one thing that in all these assays, and the number of papers that are coming out per day, I don't read them all, but when you do these assays, what's reported in a PCR assay is plus and minus usually. So you can get the value of the number of PCR cycles, and that gives a lot of information, but that's almost never reported. So for these people who are negative and positive and negative, you wonder if to me, 35 is a number of cycles that I consider positive in my lab. Clinical labs go up to 37, which I find really low. So, but if you're dealing with ones that are hovering around 35 or 36, you could easily imagine fluctuating on one side or the other. So I don't know if this is the explanation. But
so you may be interested. Christian Drosten told me that in the early Munich cohort in Germany, they had one patient who had a CT of 13.
That's a lot. Yeah, that's really, really high, because I think the reports now for people who have the lower respiratory tract disease and are severely ill have usually values about 20 to 21. So 13 is incredible.
Yeah, he was... and he was very mildly infected, apparently. So that's a kind of person who could really transmit well, I guess.
Yeah, that's what you'd worry about. Exactly. That person, for some reason, is completely tolerant to the virus. It's sitting up there in his nose, replicating just fine, and he's not even seeing it. His immune system's not even seeing it. Yeah.
There is the other aspect I wanted you to discuss with us is the really severe disease. Can you tell us what are the components of that? Because we know there's partial virus destruction of alveolar cells, and then there's an immune component. Can you take it apart for us?
Yeah, I think that to my mind, I think the major part is the immune component, that you get a combination of by having a virus there that's causing some destruction, you have a huge influx of cells and cytokines and molecules in the blood that cause those cells to come in. And these cells and cytokines actually destroy tissue even more so than the virus does. So that a lot of our old studies with coronaviruses showed you could have an animal with bucket loads of virus, and without an immune response, the mice were just fine. So I'm a strong believer in thinking that the immune response is actually causing a lot of the disease. Mm-hmm. And we've done things with SARS and MERS too, where if you negate parts of the immune response, you can now actually take mice from being very sick to being very well without changing the amount of virus particularly. So, and I think it's what exactly that means is not 100% defined, because we know there's a group of these cytokines, and neutralizing or taking away one may not have much of an effect, and lots of different cells come in. The ones we most worry about are these macrophages again, and sometimes neutrophils, which are largely for bacteria, but can also come into the lungs in a severe viral infection. And then they release other molecules that cause destruction of the lungs, and you end up with the situation where you can have what's called the acute respiratory distress syndrome, which is a kind of end phase to, and not necessarily end because people survive it, but it's a kind of end phase destruction of the lungs, and it'll go on itself even if you get rid of the virus. So I think that the host does a lot here to make the infection be bad. And that's why when people talk about therapy, the idea that you need to both eliminate the virus and do something about the immune response is probably the way to think about it, because if you do one without the other, you can have a problem.
And this was a characteristic of SARS and MERS as well, right?
Yes, I think so. That's right. I think it was a characteristic of SARS and MERS. In fact, SARS, the thinking was the same. So people were being treated with a drug called ribavirin, and then were treated with steroids to eliminate the immune response, or to tone down the immune response. So ribavirin was supposed to eliminate the virus, and the corticosteroids were supposed to tone down the immune response, and so the patient would slowly get better. The problem with the approach is it turned out that SARS was resistant to ribavirin. So you were toning down the immune response without getting rid of the virus, without clearing the virus. So this is a problem.
So there are people being treated with, say, anti-IL-6 receptor antibodies, and we don't actually know the outcomes yet, but it's been suggested that this may be helpful. But you would say you'd need more than just that, right?
Well, maybe not. Maybe this is because what I like about this approach, and I don't know that IL-6 is the right target. There's some hints that it is, but no one knows. Steroids knock down everything. So steroids was such a good immunosuppressive agent that they really didn't allow anything to still go on. It's sort of like so they didn't allow any clearance to occur, or they minimized it so much that people ended up with worse disease. Yeah. But there's other cases where, for example, neutrophils, which come into the lungs in some viral infection, we think they probably contribute to worse disease. These cells are very potent for clearing bacteria. They make a lot of compounds that are very effective at killing bacteria and also cells. So we think that if you eliminate all neutrophils coming into the lungs, that patients will do worse because there's a role for them. But we think that, so the ideal thing would be to cut down the number getting into the lungs. So if IL-6 is just one of several factors, or is even a main one, but there's lots of other subsidiary factors, and they all still remain, then you can easily imagine a scenario where you improve the ratio of host damage to virus clearance, and now the patient does much better. Because you're still clearing the virus, you may still have some damage, but the goal is to not have the severe damage that either results in death or very severe lung damage.
So when we see this immune enhancement of diseases, is by that time the virus level, is that dropping already?
Yeah, that seems to be a characteristic of all these coronavirus infections, and certainly what we saw in mice. In mice, of course, it's easier to see, where you can see peak levels of virus occurred, and mice weren't terribly sick, and then they got sick just when the immune response came in and started clearing the virus from the animal. So it seems to be a characteristic of these viruses, as much as we can tell, that virus infection is the clearance of the infection itself is what causes disease. This is kind of the opposite of Ebola, where the virus titers seem to increase during disease, and that's what caused poor outcomes.
I'm often surprised when I hear people talking about the important role of IL-6 here, because I usually think about interferons as being such important cytokines in many of these viral infections. Is there any role for interferons in this disease that we're seeing?
Yeah, so we have interferons is certainly a marker. We did experiments in mice showing that in some cases the interferon all by itself caused more severe disease. If you eliminated the interferon and completely eliminated it, you actually took a mice that died from SARS into one that survived. So huge effects of eliminating interferon. And we know that if you give interferon early, that it's always good. So if you give interferon before an infection starts or just as it's starting, it almost always results in faster virus clearance, much lower host responses, much less tissue damage, and much better outcomes. If you give interferon late, we did studies that showed you actually could make things much worse, even in mice that were getting better. In people, this may be relevant for people. Of course, I don't really know, but there are studies using interferon in patients who have MERS. And the studies basically were inconclusive because some people got better and some people got worse. And I wondered if this wasn't because some people were getting interferon early in the infection, so preventing bad host responses, and others were getting it when the virus was clearing, and so made things worse. So and this is, of course, the thing with humans. It's hard. So IL-6 may have a role a little later than interferon, and may not. Without having magic ways to look in people. But hopefully there'll be a biomarker that'll help us sometime in sorting this out. But otherwise, it's really people just making the best decisions and doing controlled studies, which of course is a key thing that's often not done now in the context of COVID-19.
Speaking of mice, I assume people are trying to make a mouse model, right?
Yes.
And is that going to involve beyond just putting ACE2 in, right?
Well, yeah, probably. So there's a lot of approaches here because we had made a mouse model for SARS. We made a mouse model for SARS where we took human ACE2 and put it into mice. And we, at the same time, other people were adapting the virus to grow in mice. So you didn't have to use any special mouse. So the problem with our approach worked very well. I remember the first time we infected mice with it, they all had bad lung disease, and we thought, wow, this is terrific. Well, it turned out that they had bad lung disease because they got something that mice normally never get, which is they got an overwhelming brain infection and they aspirated. So that means that they weren't able to control their airway anymore. So food was going back from the stomach into the airway. And that's why they had lung disease. So not what we wanted. And so these mice, the ones we had, and then we froze down the sperm from the mice in 2009. And then when this came about, we donated the whole system to Jackson Labs. And that's the mouse that they're actually resuscitating and should be available next month or the month after for large groups. We worry about this mouse because we worry, well, will it get brain disease? It'll still be useful for lots of things, but what we've done since then, and others have as well, we're not the only ones, so we've taken so mice are normally not infectable by the virus, but we changed the mouse ACE2 just with one amino acid that should make it infectable. So those mice will be ready in the next month or two as well. And we probably will have to do at that point what you said. We probably will have to take the virus and adapt it to the mice so it causes more severe disease. The other way you can do this, because it only took one amino acid to change the human ACE2, is other people have taken the virus and modified that so now it can ignore or can compensate for the one amino acid that's not right in mouse ACE2. So that's another way of making a mouse model. So I'm sure that by the early fall, we'll have lots of mouse models that'll be useful for studying why the virus causes disease or pathogenesis, studying why, which antiviral agents might be most useful, and for assessing vaccines. Vaccines, of course, may move faster than that because nobody wants to wait until October to start testing vaccines. They're already being put into humans. So it may not be so useful for that, at least in the short term.
So, Ralph, I remember that. Sorry, Stanley, not Ralph.
That was the person who developed the mouse. That's great.
Right. That when we talked to you in Iowa, you said that you had a longstanding interest in neurotropism, and that in the natural SARS and MERS infections, that there is some evidence of that in humans. And is that also the case in SARS-CoV-2?
Well, in SARS-CoV-2, it's almost more exaggerated, maybe because there's so many cases, but now there's several reports of people having neurological disease or psychiatric disease. And then people have done MRI scans of their brains, and they're seeing abnormalities. I'm not sure. I can't think of how many cases actually show virus in the brain. I think there may be some on autopsy. I know in SARS-1, there was at least one report showing virus in the brain in people who had died. So it wouldn't surprise me if this virus invaded the brain sometimes. It's not very common. With SARS-1, of course, there's 8,000 cases. Here, when you have whatever, we're up to almost 2 million, you're going to have such a huge denominator that not that many cases will still be a large number. The other thing I think that's more interesting that's of course interesting, but what's really more curious is this whole concept of losing your sense of smell. Because that seems to be a common finding that I hear from lots of people who have really mild disease, and I guess they can go on to have more severe disease too. But people have lost their sense of smell, and then it takes about a week or 2 weeks to come back, and a lot of them come out to be COVID-19 positive. So what exactly is going on here is really almost counterintuitive, because you would think that if you're having the virus mucking up the nerve endings in your nose and causing not much else, that would be exactly the situation where you would see some sort of brain infection. But it seems to be not at all, because those people have subclinical disease for the most part. But what's going on there, I think, is really an interesting question.
And do people recover their sense of smell?
Yeah, in a week or two, usually.
Okay.
This also occurs with other viral infections as well. So it's not totally novel here, but I think what's novel about it is in the month of April, where we have so many cases of COVID-19, it's likely that anything we're seeing has something to do with COVID-19. There's still flu around and other pathogens, but we're thinking this is mostly COVID-19 related.
Stanley, there seem to have been, with SARS-CoV-2, super spreading events. or at least that's what I see in the news. I'm interested in your perspective on these, in particular, whether there might be such a thing as individuals who themselves are super spreaders, or whether these events have more to do with the circumstances surrounding an infection, like a crowded place over a long period of time.
Oh, yeah, I think it's probably a combination of the two. If you just go back to the patient that Vince was talking about, who had a huge amount of virus in their nose, you could easily imagine that that person could infect lots of other people, especially if he or she had mild infection and was barely symptomatic and was still, had some mild symptoms. And so people wouldn't have even thought about his being a candidate spreader. But that would be the kind of person. And during SARS-1, that's what we were thinking. We were thinking those people are generally sick. This would be somebody who's not even that sick, but generally sick and sneezing and coughing a lot with lots of virus per droplet. So the more likely that people who are downstream of that would get infected. So, and then of course, the more you're in a crowded room, the more people you can infect. So I think it's all a combination of events. I know in SARS-1, when we used to talk about these super spreaders or super spreading events, and there was just so few that it was really hard to study them because you almost identified them post facto. And then so you couldn't go back and say, okay, let's measure virus loads 2 weeks ago and see if that's the reason this person spread virus so readily.
There have been some reports that the virus SARS-CoV-2 can infect immune cells. Have you seen that? And what do you think?
I haven't seen that. I can't comment on it. I know that SARS-CoV-1 affected macrophages abortively, so you didn't get virus out. It could infect it, but you didn't get virus out. But it did make the cell produce cytokines and chemokines. MERS was reported to infect macrophages productively, so you actually get virus out. But I also don't know how common that really occurred. That's in human cells. In mouse cells, both of those didn't infect them at all. And then MERS may have affected T cells, but I don't know about the new virus. I don't know those reports or how good they are.
Yeah, Brianne is shaking her head. She doesn't like it.
There is one figure in one of those T cell papers that I have a lot of problems with. So.
Okay. Well, like anything else with this virus, it's all amplified, right? Because there's so much, as Stanley said, and the littlest thing you hear and people want to know about it. In terms of your experience, Stanley, you're a pediatrician. Are you seeing pediatric patients there with virus infection?
Yeah, there's some. I think there was, just the last time I looked, maybe 3 hospitalizations right now of children with COVID-19. I don't think they're terribly sick. One of them was in the ICU, but not really for COVID-19. It turned out he got a febrile seizures because little children often get seizures associated with fever, and that's all he had. So he also had COVID-19, but once the febrile seizure was gone, he was back to being a fairly mildly infected child. So I think there's cases I think all these diseases are really interesting in the sense that they don't infect children that much, both SARS-1, MERS, SARS-2. So children get mild disease, and SARS-2, we have information now that they're infected at the same levels as other age groups. They just don't get sick. With SARS-1 and MERS, we didn't know that for sure.
That's a great strategy for a virus. Just infect kids who are naive immunologically, don't cause disease, so nobody bothers with it.
Right, exactly. And also, and spread the disease happily to other children in child care centers or to their parents or friends or whatever.
And then the parents don't get sick because they have a little bit of immunity, right?
Right.
It's a good strategy. Do you have any sense of? So our talk about antibody and immunity is interesting. So everyone's talking about reaching a level of population immunity to get the herd immunity, but do you have a sense if we will reach that, or is it possible even?
I think it's possible, and it really depends. That's why people talk so much about testing, because we don't know what proportion of the population is actually not naive anymore to the virus, have already some immune response. And so the more we know about that, the better we can guess whether there's going to be herd immunity. One thing we know, I think people who are modelers can say, well, if a virus spreads with this much likelihood to infect other people, then you need a certain amount of herd immunity. If it's less contagious, you need this amount. If you have measles, which is extremely contagious, I think the number is 95% herd immunity. So it depends a lot on how many people are actually positive and how contagious this virus is at the end.
Now, if SARS-1 had continued, would that confer some immunity to this particular virus?
Yeah, I think it confers some, whether it would, and maybe it would have been enough to make it so people, if everybody had had SARS-1, that they wouldn't have had there wouldn't have been much severe disease with SARS-2. Yeah. But, the studies so far, because when this first came out, the studies so far showed that plasma from SARS-1 immune people had offered some protection against the virus, at least in this test tube, but it wasn't great. Mm-hmm. It's certainly not compared to using the initial immune serum to the virus, to COVID-19 virus itself.
Yeah, I'm asking because I'm wondering if this pandemic will provide some immunity to a future SARS-like virus that would emerge from bats.
It's hard to know because the surface glycoproteins are always different enough, so there's definitely some divergence.
Yeah.
Thinking about the herd immunity question, it's hard for me in the current circumstances with SARS-CoV-2 to think of an alternate scenario. I just don't see this going away. It's so widespread now and sufficiently transmissible that once this first wave is over, it's hard for me to imagine that the virus is gone, just not being transmitted very well. And I see it hard for me to imagine a circumstance where it's not ultimately going to spread throughout everybody. Am I making sense?
Yeah, that's kind of what I think too. And that's why knowing how many people are actually infected. So, all these people who have mild disease and never saw any healthcare worker, you wonder how many of them are out there. Is it minor? Is it a small number or is it a big number?
I hope it's a huge number.
Yeah, that's right. So if it's way off, that's what I hope too, that it's a huge number. And so people are much protected and herd immunity will cut in and there'll be a second wave, but maybe it won't be so bad. Certainly we know from the flu in 1918, that wasn't the case. The second wave and the third wave were worse than the first wave. So it's not predictable, but we know so much more now about how to think about these things. Even if we don't have antivirals and we don't have vaccines yet, we understand how the virus is spread much more.
People are talking about antibody tests. So most of them seem to me are going to be rapid, 15-minute, non-quantitative. Is that going to be useful, you think?
Yeah, I don't know. Because you so much want to know, are people protected or can they transmit? And these kinds of tests, our work with MERS is really jaundiced me because that's the right word. Because we found the commercial test wasn't as good as our neutralization test. So in Saudi Arabia, we'd have 20 positives by neutralization. One of them would be picked up by ELISA. Oh. So it's I'm a fan of making sure we get it right. But how to do that exactly? The number of calls I get now about asking for the cold coronavirus just to make sure there's not cross reactivity. But it's all not easy. The cold coronaviruses, 2 of them grow poorly, and the other 2 grow well, but at least until recently, ATCC has been out of them. So it's been a challenge for people trying to do it right and get their antibody testing working well.
Stanley, on a different topic, people have said or worried that healthcare workers maybe get a worse disease because they get a higher dose of virus. And so we've had kind of a raging debate on the podcast about systems perhaps in mice where you could test that. And so I'm wondering if for the mouse hepatitis virus, if there are studies where a low dose and a high dose are given and if the disease is different. And I don't just mean more severe, I mean different sort of in the constellation of clinical signs.
Yeah, so no, we only see a quantitatively different disease, not a qualitatively different disease. So we don't see that. If we give a lower dose, or whatever we're looking at will be less. There'll be less of it. If we give a higher dose, there'll be more of it. But the one of the challenges, of course, you give a low enough dose, it's almost like vaccinating. So you end up with mild disease and good immune responses. And it's only when you cross the line where the virus is growing a little too quickly for the immune response that you start seeing disease. So we don't see, we haven't modeled that in our mice thus far. Because, the other thing about healthcare workers is, so not only are they exposed to larger amounts potentially, they're exposed to aerosolization if things are not done right, which other people wouldn't be, during intubations or obtaining trach aspirates. And then the third thing is, at a time like this, working in a healthcare setting is so incredibly stressful that your basic metabolic milieu is different. So I think all those things could contribute to healthcare workers getting more severe disease.
So that's correct that there is an observation that healthcare workers get more severe disease? Yeah. Okay. But I think that's a good point. There are a lot of things that could be contributing to that. So how do you see the vaccine trials panning out? Are we going to have, I don't know when phase 2 will happen, but it's conceivable that there won't be a lot of circulation by that time, right?
Yeah, well, in China, they're ready. The vaccine they're putting in phase 2 trials already.
Is that the adenovirus vectored vaccine?
Yeah, which one might think would have some issues, but that's already in phase 2. And the RNA virus, I don't know how long that'll take to get into phase 2.
Well, that trial is supposed to end on June 1. Okay, and
really?
I figure,
phase 1?
and I figure that they would have results from that almost instantaneously after that. And what's not clear is, to me is, I don't think there would be any problem recruiting for a phase 2 or funding it under these circumstances, but I don't know what sort of, how fast the paperwork would go through to initiate that. But I can see that, happening, well, probably pretty quickly after the first trial is done in June.
Yeah, that's what I would guess too. And in fact, the concern is, especially seeing on this vaccine enhanced disease committee, is that we're going to miss some vaccine enhanced disease that's more concerning than what we've talked about. Where you have changes in what the cells look like in the lungs, but not much else. So this is a real important issue. Of course, it matters because this country is so polarized about everything, including vaccines. The last thing you want is for the vaccine to have an issue and have nobody get vaccination and then having the disease be much worse because of that.
Yeah. Can you, based on what we said earlier about the pattern of infections with the common cold viruses, and maybe SARS-CoV-2 will eventually be similar. So let's say one or multiple vaccines are successful, they're licensed, and they're used. Can you see a time when we don't need them anymore because we don't vaccinate against the common cold coronas, right?
Yeah, that's the same story, right? That's the same thing happened with smallpox, where we don't vaccinate anymore. So you could imagine that happening with time.
Yeah.
It would be a wonderful outcome if we got to that point.
Yeah. It would be very ironic because people want a vaccine right now, right?
Yeah, that's the way it always is. But that's why vaccine companies are always suffering because on one hand, we wanted the vaccine yesterday, and then you start doing it in smaller number of patients, and there's more side effects, some real, some not real, and it ends up being a mess.
By the way, I'm sure everyone knows this, especially Kathy. Sunday was the 65th anniversary of the licensure of inactivated polio vaccine. Mm-hmm. And it took almost 50 years to make that vaccine.
Yeah.
From the year it was isolated in 1906. Really remarkable. I have one last question is, what do you think about the fact that this virus, SARS-CoV-2, has a different name from the disease, COVID-19? Does that make sense?
I was on the committee. Yeah, I was actually on the committee that named the SARS-CoV-2, and I was at a thesis defense in the Netherlands, talking to the guys who really were adamant that it had to be named this. Mm-hmm. And this was just an impossible discussion because they felt the people who knew the genetics of it said, this is SARS-CoV-1. We can't name it anything else. It's too close. But then some of us who saw patients and people from people from China said, this is ridiculous. You can't put the word SARS in there because that's going to put people off. So, and about the same time we were having our meeting, then there was a WHO meeting, and right before the meeting, the committee at the WHO, which I don't think included any coronavirologists, named it COVID-19. And so we at the meeting, I had to leave early, but the other people who were still there said, this is really not a great idea. So now you can call it whatever you want. You can call it SARS-CoV-2 disease. You could call it the COVID-19 virus. You could call it the COVID-19 disease caused by the SARS-CoV virus. I mean, it's a mess. And so I think in the world, they're going to call it, and then other people, because it's so confusing, still call it the novel coronavirus. Yeah. Then people, as we know in this country, people call it the Wuhan coronavirus. Unfortunately, that's become pejorative, so that's a bad idea. But it is called so many names because it's not something simple.
But SARS is the disease, right? It is SARS.
Yeah, but SARS coronavirus is a virus.
Yeah. But so SARS-CoV-2 is okay, don't you think?
SARS-CoV-2 is for a virus. The only problem was that people thought, boy, SARS has such a bad connotation. We don't want to make this the same.
But I liked SARS-CoV, the original, because it had both the disease and the virus together, right?
Exactly. That's what we all liked. And the MERS-CoV we had the same issue, but people decided after that they weren't going to name a disease after a place again because it sounds like it contaminates the place. Just like in the beginning, people were talking about the Wuhan or the China virus. It's very, very important that it was never named that because it's spread around the world and who cares where it started. And you end up doing blame instead of anything useful out of it.
I just don't like COVID. I think that's very, coronavirus infectious disease. What does that mean, right?
Well, I try to tell people that are non-scientists that, because I think they pretty much understand it, that there's AIDS and then there's HIV. Yeah. And so we have COVID-19 and SARS-CoV-2. But I know of someone who overheard some nurses talking about COVID-19 and saying, well, yeah, they named it that because don't you remember before there was COVID-18 and 17?
Oh my gosh.
So, yeah.
But the naming of HIV was a mess in the beginning too, right?
Yeah.
Mm-hmm.
Yeah, that's right. Remember that? Yep. What was it named originally? Something.
GRID.
GRID and HTLV3 and LAV.
Yeah.
That's right.
Yeah. And some of those were worse than others.
All right. That was great. Thank you. This has been TWiV 602. You can find it on any good podcast player, any podcast player, period. The show notes are at microbe.tv/TWiV. Send us your questions and comments, twiv@microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Our guest today from the University of Iowa, Carver College of Medicine, Stanley Perlman. Thanks so much, Stanley.
Thank you for having me.
It was great. Really explained a lot. Brianne Barker is at Drew University over on Twitter, BioProf Barker. Thanks, Brianne.
Thanks. This was really useful. I learned a lot.
Kathy Spindler's at the University of Michigan, Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun. And sorry, Stanley, I got here a little bit late, but I'll be interested in hearing the whole thing from the beginning later.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. And Stanley, great to see you. Great to talk to you.
Good talking to all of you.
I'm Vincent Racaniello. You can find me at virology.blog. I want to thank ASV and ASM for their support of TWiV, Ronald Jenkins for the music. This episode was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.